Welcome back. Today we're talking about DNA structure. And DNA is a very important molecule that is required for all cells to be able to live. It's our molecule that allows us to pass down genes from one organism to another. And so today's essential question is basically, what are the chemical components of DNA and how are they connected? And so if you remember back from first semester, we should have learned that DNA is a macromolecule. And what macro means, remember, is that it is a large molecule made up of smaller subunits. So DNA's subunit is called a nucleotide. Subunit is a nucleotide. Each nucleotide is going to contain three things. The first thing it's going to have is a 5-carbon sugar. And that 5-carbon sugar is a part of the name. In DNA, we call it deoxyribose. The second part is called a phosphate group, and this is the same phosphate group that we found in ATP. And the sugar and the phosphate groups are going to make the backbone, what we call the backbone of the DNA molecule. The last part is kind of what will be the rungs of the ladder. Each nucleotide is going to contain one nitrogenous base. And there's 4 types of nitrogenous bases that we can have. We can have adenine, thymine, cytosine, or guanine. So let's take a look. Each nucleotide is going to have one deoxyribose sugar, a 5-carbon sugar, a phosphate group, and a nitrogenous base, either adenine, guanine, cytosine, or thymine. Now, these can be joined together. Each nucleotide can be joined together in any order. And that order is going to be based upon the previous cell or who our mother and father were. All right, but they can be joined together in any order. We could have 10 adenine nucleotides all in a row, followed by 10 cytosines, followed by 2 guanines, and 3 thymines. It doesn't matter the order. The order is going to be significant when we're making proteins. So each nucleotide is made up of a phosphate group, a 5-carbon sugar called deoxyribose, and a base, a nitrogenous base called adenine, guanine, cytosine, or thymine. Now, this molecule, this is not the whole molecule here. DNA is also going to be double-stranded. So DNA is double-stranded. Meaning, on the other side of these bases, we're going to have another nitrogenous base that matches up, connected to a sugar, so I'll put an S there, and then a phosphate group. And so we're going to continue connecting these things on the other side, and it's going to look kind of like a ladder. The other thing that we know about DNA is that it has a helical structure. And so we should have looked up the word helix previous to doing this lesson. And all a helix means is that we're going to twist around a single axis. And so we've got this double-stranded molecule that's going to twist around the same axis and kind of create this twisted ladder. All right, so how did we figure all of this out? And actually, the story is pretty interesting. I'm going to just kind of hit on some highlights, but this was a structure that we knew it existed, but we didn't know what it looked like for a long, long time, and it was kind of a race to see who could figure out the structure of DNA first. And somebody that was very important in figuring all of this out was a man by the name of Chargaff, and he created what we call Chargaff's rules. What Chargaff did is he counted all the nitrogenous bases, all the different types of nitrogenous bases. So he counted all of the bases. So he counted how many adenines there were, how many guanines there were, how many cytosines there were, and how many thymines there were. And what he found was the percentage of adenines in a sample of DNA roughly equaled the percentage of thymines. Also, he found that the percentage of cytosines roughly equaled the percentage of guanines. And so Chargaff's rule states that adenines will always bind with thymine. And so we see that here in this molecule. We have our adenines bonding to thymines, thymine with adenine. They will always go together as a pair. He also found that cytosines will always bond with guanines. All right, and we see that as well. We've got a cytosine here bonded to a guanine. We've got a guanine bonded to a cytosine. And so we have this ladder structure with our rungs being the nitrogenous bases, and those nitrogenous bases are going to be linked together in a very specific way. The next person who's really important to us is Rosalind Franklin. Rosalind Franklin worked at the University of Chicago, and she was a chemist. She was a biochemist, and she was very skilled in what we call X-ray diffraction. And basically she took X-rays of different molecules to see once what they looked like. And from those X-rays, we can determine some of the structural attributes of those molecules. And so this is an example of Franklin's X-ray diffraction photograph of a DNA molecule. And so Franklin discovered through X-rays, the X-rays showed that the molecule was twisted. In other words, she discovered that DNA was a helix. And so that's why I have these pictures over here. We have our three different kind of pictures of DNA, each one showing that we have this helical structure, this twisting staircase or this twisted ladder going around. I could put a bar right through the middle here. We can see how it's twisted along one axis. This is the same molecule, just a little bit different representation. But we had that ladder that's twisted, and we found that out because of Rosalind Franklin's X-ray diffraction. The people that are credited with finding the structure of DNA are men by the name of James Watson and Francis Crick. And their pictures are shown here on the slide. Watson and Crick really found the structure of DNA. So they discovered the structure. And they did it by building a model. In fact, they built several models to kind of figure all of this out. And they took information from several people, some that I have named, some of them that I haven't, but they took Rosalind Franklin's pictures and they took what Chargaff found out, and they basically put it together with some chemistry that they figured out and by building models. And one of the first things that they said was, we have this ladder that's twisted, and the ladder has the nitrogenous bases. The nitrogenous bases are on the inside. And so they're making our rungs of our ladder. Previously, people had thought that these nitrogenous bases were kind of sticking out like porcupine needles, and so they discovered that they were in the middle. The next thing they figured out was that those bases are connected with hydrogen bonds. So they found out these were connected with hydrogen bonds. Hydrogen bonds are very weak bonds. And because of their weakness, we can basically unzip this molecule without exerting a lot of energy. And so eventually they said, this shows how we can copy DNA from one cell to another. The third thing they figured out by building their model was that the strands, our backbones, go in opposite directions. So we can see that this one is kind of going in this direction, and this one is flipped upside down and is going this direction. And so they said that the backbones, or the sugar phosphate backbones, are anti-parallel. Taking all this information in, building up their models, allowed us to find the structure for DNA, which then allowed us to make some hypotheses and some theories about how cells copy the DNA before they multiply, and how we can actually make proteins. All right, and so here's a picture of Watson and Crick with their model of DNA that they built in their office. And I hope this video helps a little bit on understanding the structure of DNA.